right. So I'm so delighted to be here today. And I have entitled my presentation today, The Sound of Silence, Silencing the HIV Reservoir, New Opportunities with Metformin. And I understand that this is a webinar co-sponsored by multiple collaboratories, HOPE, DARE, Rid HIV, and CRISPR for Cure. I, of course, represent the HOPE Collaboratory, and then, as was kind of discussed in the beginning, we are kind of known as the Block and Lock Collaboratory. And this is, one of the photos from one of our recent annual meetings, and all the folks here are, in some sense or another, working on this goal of trying to achieve block and lock. But one point I want to make is that, it's not the case that all these collaboratories are working in silos. There's actually a lot of interactions and crosstalk between the collaboratories. And just as an example, I'm actually very much integrated with the DARE Collaboratory. It's the other one that also is based in the San Francisco Bay Area. And in fact, Steve Deeks, one of the heads of the DARE Collaboratory, calls me and others a FOD, F-O-D, a friend of DARE. And in fact, I even made it into their annual photo right here. See, that's me. So anyway, a lot of crosstalk between the collaboratories. Really, really fun to interact, cross-collaboratory work. I thought to begin with, I would just tell you a little bit about myself, because I don't think I've met all of you. So about me, I was born in the Bay Area of California, specifically in Berkeley. But I actually grew up all over the world, including West Coast, East Coast, Asia, Europe. But my high school and college was in the Bay Area. And then after finishing college, I decided to go to graduate school in Boston. And it was during those years that I began studying infectious diseases and became really passionate, specifically actually about women's health. I was studying immune responses to chlamydia trachomatis, which is a common bacterial sexually transmitted disease. Now, it was during those years that I became fascinated by CD4 T cells, because I was studying the interactions between CD4 T cells and chlamydia. And I also knew, of course, that CD4 T cells were the main targets of HIV. And so I was kind of, hadn't studied HIV up to that point, but became very interested in it. And then, so after I finished graduate school, I decided to do a postdoctoral fellowship to begin to study HIV. And I worked with Warner Green, who I think many of you know. He's a prominent HIV virologist. And that's when I began studying HIV and became really passionate about the topic. And I have continued studying HIV ever since. It's now been almost 20 years that I have stayed on at Gladstone and UCSF doing research with an HIV focus. Okay, so I want to begin by talking a little bit about the community efforts at the HOPE Collaboratory. Community is extremely important for HOPE. Here are some words of what community means for the HOPE Collaboratory. And here are some nice photos of various events. So this is a community event, a HOPE community event. Raif, who's here, is right in the center. And down here, this is actually a visit that a group of us did to Florida. And what we are showcasing here is actually a piece of art that was created as part of the HOPE Collaboratory. And actually, it's a piece of art illustrating the notion of block and lock, which I will say more about in a bit, exactly what that means. But basically, it's these chess pieces, and I actually am not a chess player, so I don't actually understand it, but it's these chess pieces where essentially this one piece is blocked and locked so that the piece has no choice but to stay put. And so that is the idea of block and lock. Now, as I mentioned, HOPE is very busy with community efforts. The community efforts are led by Patricia. And I borrowed this slide from Patricia, and it really just goes to illustrate how busy she and the rest of the HOPE Collaboratory community folks have been. So this is just a period of less than 1 year. This is last year. And you can see in just less than 12 months how many community events the HOPE Collaboratory has put together. So really just highlights that we are doing a lot in the context of community. And one thing I want to point out is that some of the stuff that we're doing very much engages closely with Last Gift. And in fact, Last Gift is part of the HOPE Collaboratory. Now, I think probably all of you here have heard of Last Gift, but in case you're not familiar with it, it's a cohort led by Davey Smith and Sarah Gianella based at UC San Diego. And it's a cohort of altruistic individuals who choose to essentially donate their entire bodies for HIV research towards end of life. because it's really important to understand what happens in tissues, and it's really, really hard to do that in living individuals. And so the cohort is called Last Gift because this is these individuals' last gift for the community. And, recently there was a documentary, a beautiful documentary put together about Last Gift. It's called The Last Gift, Jim's Courage, Our Hope. And it really talks about one Last Gift participant, Jim. It talks about Jim's legacy. This is actually Jim, and this is his wife, Susan. And so the documentary goes from the period when he is interviewed about why he decided to enroll in Last Gift and what that's like, all the way to the time until after he had passed. And then the documentary also interviewed Susan. And actually, my lab in the Gladstone Institute is a part of this documentary as well, because we showcase some of the research that we were able to do and what we could learn from the gift that Jim had provided. And actually, this documentary, excitingly, was it was announced just about a week ago or so that it was nominated for an Emmy. So that's very exciting to see. So if you haven't seen the documentary, I highly recommend it. Definitely put me to tears. For everyone else I've talked to, and also put them to tears as well. So just as a warning. The other kind of Last Gift update I want to provide is Loreen Willenberg. And so I think everybody here knows who Loreen is. She's an amazing person. She is one of two known what's called exceptional elite controllers. She also has been nicknamed the San Francisco patient. And essentially, her body could naturally eliminate HIV without antiretroviral therapy. And so she's very well known in the HIV field. She's also an amazing person. She's a talented landscape artist, but she was also very dedicated to HIV research. Now, she knew about Last Gift, but she isn't based in San Diego. So all Last Gift participants are based in San Diego, but she really wanted to essentially give this last gift as well. And so she last year got in touch with Sarah Gianella, and started discussing what are the possibilities of actually enrolling in Last Gift, even though she is up in the Northern California, not down in San Diego. And so, Sarah's team reached out to my team, and we had quite a number of meetings last year planning for how we would actually enable Loreen to enroll in Last Gift. And as many of you know, Loreen sadly passed on April 6th, just recently. But we made it happen. She enrolled in Last Gift, and essentially within hours of her passing, Sarah's team got on a flight and flew from San Diego to Sacramento, where Loreen was. There was a team from my group who immediately drove there, and they essentially arrived at the same time as Sarah's team, did the rapid autopsy, and we have an amazing collection of tissue specimens, some stored in my lab, some in Sarah's lab, but this will really be essential for understanding how it is that someone can naturally eliminate HIV. So I think this will be an amazing gift from Loreen. Okay, so I'm going to now move on to the scientific program of HOPE. As I mentioned, HOPE focuses on this block and lock notion of HIV cure. And so this is a, visual representation of block and lock. You don't need to kind of focus on what all these little symbols mean, but, just at a high level, what it focuses on is this notion that the HIV reservoir, which is the main barrier to an HIV cure, is in fact actually not silent. It's very active. It's producing HIV gene products. And the idea is that we can convert this active reservoir into one that is silent, ideally permanently silenced, then we can achieve HIV remission. And so the idea is that we can somehow, for example, using some drugs, convert this active HIV into a block state, right? So where the HIV is no longer active. But the idea is you don't want to just, treat individuals with some drug that constantly blocks for their entire lives, but rather you want to temporarily treat with some drug, remove the drug, and then have it still be silent. And that's the lock notion. So block and lock. Okay. Now, is there any reason to think that we could actually achieve this? And indeed there is. And I think Melanie covered some of this in one of her previous presentations to the group. But in fact, our DNA, our genomes are actually littered with what's called endogenous retroviruses. So these, at some point, in earlier time, like millions and millions of years ago, our ancestors got infected with a virus that is kind of like HIV. But then over these millions and millions of years, these viruses essentially became silent. They are a permanent part of all our genomes, but for the most part, they don't cause any harm because they are silenced. And in fact, in some instances, they actually have benefits. So that's kind of a different topic, but for the most part, they are silenced. And they are silenced because there are these proteins called crab zinc finger proteins, represented here. This is shaped like a crab because, it's KRAB, but that's the notion, crab. These crab zinc finger proteins will silence these endogenous retroviruses so that they are not active. Okay, so there's already plenty of examples of this occurring in nature. And so the idea here for the HOPE Collaboratory is that, HIV is not an endogenous retrovirus. It's what's called an exogenous retroviruses. But can we accelerate evolution in some sense and convert it into something that would resemble an endogenous retrovirus by silencing it? So that's why we're trying to learn a lot of basic biology about how these crabs here are actually silencing these endogenous retroviruses. And so, how would we silence HIV? And so as a collaboratory, we like to think of, trying to identify what we like to refer to as SPAs, because it has kind of a nice sounding name, but also the idea here is that these are drugs that are silencing promoting agents, so SPAs, right? And the overall goal here is that we would convert an active reservoir via these SPAs into first a block state and then eventually a lock state. Okay, so what I will talk about today is actually a study that we recently published. This is the study. It has kind of a long title, but what I want to draw your attention to is essentially what we had done is we identify ways to silence HIV, both from the outside as well as from the inside. So that's what this means when it says cell extrinsic and cell intrinsic. Essentially, if this is a representation of an actual HIV reservoir cell, we identify genes from within, intrinsic, that could silence HIV. And actually, one of the genes can be induced by metformin. And I'll say more about that, but that's where the metformin link comes in. But just to also point out, there are things from the outside as well. These are immune cells called NK cells and CD8 T cells. You can think of them as soldiers that patrol around the immune system and they can recognize infected cells. But there are also features from the outside that can silence HIV. But today I won't talk about that. Instead, I'll just talk about these internal or intrinsic factors. And so this is kind of, a more scientific way of presenting what we found in this study, but I actually like this image as well. So this is the notion of the cell intrinsic and cell extrinsic kind of block and lock for HIV. So here's HIV. It is in jail, but there's two levels of locking. Okay, there's these chains around the HIV, and these are the intrinsic aspects. That's these genes that I will talk about, one of them induced by metformin. But there's actually another layer of locking as well, and it's these soldiers that I mentioned, the CD8 T cells, the NK cells are also able to provide another level of lock. Okay, so the plain language slide of what I'm going to talk about today. So what was the key question that was asked? It was, can we identify new ways to achieve block and lock by analyzing samples from people with HIV who underwent analytical treatment interruption? And the answer is yes to that. What was the key finding and take-home message? Well, by analyzing CD4 T cells from those undergoing treatment interruption, we identify new targets for achieving block and lock. Furthermore, metformin could potentially help achieve this effect. And finally, how is this important for an HIV cure? Well, metformin is a safe and affordable drug already used by many for diabetes and other conditions. Okay, so the overall goal of our study was to try to identify predictors of HIV rebound upon treatment interruption. And so I think most here have seen this kind of general curve where that antiretroviral therapy, or ART, will suppress HIV replication to below limit of detection. But of course, the problem is that when an individual goes off of therapy, typically the virus rebounds really soon, within just a period of a few weeks. But that's not the case in all individuals. There are some individuals that rebound much later, and then there are other individuals that rebound much, much, much later, right? And we call these post-treatment controllers. Some of them don't even rebound. And, as a field, we still didn't have a good understanding of, what determines when the virus will rebound. Are there predictors at this stage that we can predict whether someone will rebound soon or much later? And so that's why we decided to pursue this study, which is called the Biomark study. And this study was led by two former scientists in my lab, Tongcui Ma and Ashley George. They have both moved on to other things. So Tongcui Ma returned to her home country in China, and Ashley George actually just recently started her own lab at the University of Nevada in Reno, where she will continue studying HIV, but in the context of women's health. Okay, so the overall goal of the Biomark study was to identify predictors of time to rebound upon treatment interruption. And so Biomark actually consisted of 4 cohorts. 3 of them were from Denmark, and it's what is known as interventional cohorts, meaning these individuals received a particular intervention, detailed here, prior to undergoing treatment interruption. And the 4th cohort is a non-interventional cohort. It's one called ACTG 5345. And these individuals did not receive any intervention and then underwent treatment interruption. And we obtained blood specimens from these individuals at the time point immediately preceding the treatment interruption. And we performed a lot of experiments with these specimens. And essentially, we were trying to see whether we could identify features of these blood specimens that could predict when the virus would rebound upon treatment interruption. And so, as you can imagine, we generate, lots of different data sets with this, but today I'll just focus on one of them, and that is just this cohort, the non-interventional cohort. And then specifically there, what we did was we took these blood specimens and we purified CD4 T cells out. So these are the primary cells harboring the HIV reservoir. And we performed a particular assay called RNA-seq. You can essentially look at thousands and thousands of genes all simultaneously. And essentially, we wanted to see which genes would predict a longer time to rebound. When someone has more of these genes, essentially, they would take longer to rebound. Okay, so what did we find? So this is the actual data from that RNA-seq analysis of CD4 T cells. Now, each of these dots represents a single gene that we identified. Okay. Essentially what is on the right-hand side are genes that are associated with longer time to rebound, and things that are on the left-hand side are associated with shorter time to rebound. And so you can think of the things on the right-hand side as quote unquote good genes. The more one has of these in CD4 T cells, the longer it takes for the virus to rebound, and the opposite for the ones that are on the left-hand side. Now, in terms of the y-axis, essentially, it's just a statistical measure. Things on the upper half are more statistically significant. You can think of them as more believable, if you will, and things on the bottom half are less statistically significant. And so we were most interested in the things in the upper right-hand quadrant because these are good genes, right? And they are the most believable ones. They're the most statistically significant. Now, remember, these data were generated from purified CD4 T cells, right, which are the primary reservoir for HIV. So we postulated that these 2 dots here, these 2 genes, may actually be mediators of block and lock, right? Because you can imagine if the CD4 T cells have more of this, perhaps HIV isn't rebounding because these genes are mediating block and lock. And just to jump to the chase, essentially, we subsequently had evidence that both of these genes that we identified were indeed able to suppress HIV. And so I'm going to actually talk about both of these in turn. I'm going to focus primarily on this first one here. And so the identity of this one, this is the top gene, the most believable one, right, most statistically significant. And it's this gene called DDIT4, which had previously never been linked to HIV before. But what is DDIT4? So DDIT4 is a gene that is activated by DNA damage and energy stress. It's also associated with hypoxia, which means low oxygen conditions. But perhaps most interestingly, it's also been reported to be a negative regulator of mTOR. And that's interesting because mTOR is essentially something that others have reported is an activator of HIV. Essentially, it promotes HIV gene activity. And so, in the science world, we like to use the symbol meaning inhibit. So the idea here is that DDIT4 inhibits mTOR. Therefore, the prediction is that when you have high levels of DDIT4, you should have lower levels of mTOR. And because mTOR is promoting HIV activity, essentially, when you have lower mTOR, you should have more silencing of HIV. And therefore, even though DDIT4 previously had not been linked to HIV at all, our prediction was that perhaps DDIT4 may be a mediator of block and lock. And so then we went about trying to test this. And so we asked the question of, okay, what if we decrease DDIT4 levels, using cells in a dish? Would that actually help to reverse this silent HIV? In other words, we can try to artificially decrease the amount of DDIT4. If it's true that it inhibits mTOR signaling, then mTOR should go up and you should have less silencing. Okay. So to test this, we worked with Zhitong Li, who was a scientist in Melanie Ott's lab. And what Zhitong was able to do was he was able to partially decrease the levels of this DDIT4 protein. Now, shown here is what's known as a Western blot. Essentially, things that are darker means there's more of something there. And what's shown in this top row here is DDIT4. So there's a certain amount of DDIT4 when you do nothing to these cells. This is the control condition. Now, under these conditions, what Zhitong was able to do was he was able to decrease the amount of DDIT4. He couldn't completely get rid of it. These are just kind of experimental limitations, but he could decrease it by about 50%. And so what he found was that under these conditions now, when you're looking at HIV activity, which we can monitor in these kinds of experiments, now when you partially decrease DDIT4, you do have increase HIV activity. So this is consistent with the notion of DDIT4 being able to participate in this block and lock effect. Okay, so far, essentially the data suggests that when you decrease DDIT4, you increase mTOR, and therefore HIV is less silent, right? So what about the opposite? So in theory, if you increase DDIT4, that would decrease mTOR signaling, and actually that should do the opposite. That should promote silencing of the HIV. And so the question then is, well, how do we increase DDIT4 levels? And so what we noticed, and initially just doing some Google searches, is that in the literature, there's quite a bit of literature out there suggesting that metformin, this really common type 2 diabetes drug, is able to increase DDIT4 levels in various cancer cell lines, cancer models, et cetera. And so this is just an example of a study that was done, not by us. This is a cancer study. This is looking at a prostate cancer cells. This is that same experimental platform I described earlier, where it's a Western blot. The darker the spot there, the higher the levels. And essentially what this group found was that when they added metformin, within 24 hours of treatment, you see a massive upregulation of this DDIT4 protein. So that suggests that perhaps metformin may mediate this effect. And that's exciting because there's a lot of info on metformin. It's a super safe drug. It was identified already back in the 1950s, and it was approved in France in the 1950s for treating diabetes. And much later, it was also approved in the US, but also more recently, there's been a lot of interest in metformin because it seems to have other beneficial effects. It seems to decrease the risk of cancer. There's some evidence perhaps that it may extend lifespan. So also in the longevity field, some folks are using it. During the COVID-19 pandemic, there's also some evidence that early treatment with metformin can decrease the risk of developing long COVID. So basically a lot of these kind of, multiple effects of metformin. Okay, but coming back to this notion that others have found that metformin induces this gene that we identified, DDIT4. This was all in cancer cells, right? But, we're interested in CD4 T cells, the primary targets of HIV. And so we decided, when I say we, I mean, I'm Kaylin Yin, a postdoctoral fellow in my lab, decided to test whether metformin could indeed increase DDIT4 levels in these T cells. And so what we did was we took tonsil cells from people without HIV. We have already access to these samples being here at UCSF because tonsillectomies are performed all the time. So we just took these cells, and then purified out the T cells. And then we did that Western blot system again, and we added different amounts of metformin. Here we did not add any metformin, and here we added a little bit, and here we added a lot more. And you can see as we add more and more metformin, we do indeed increase DDIT4 levels. So it seems to be true also in T cells, just like in those cancer cells. And so leveraging this, then what we decided to do was to test, what happens now if we treat with metformin and we look at the HIV activity, so in these experiments, we can actually expose these cells to HIV in vitro. And so what we did was we took these tonsils, purified the T cells from these specimens. And remember, these are samples from people without HIV. And we had two conditions. We either exposed to nothing or we exposed to HIV. And we did this in the absence or presence of metformin, just to see how is the activity of HIV when we add in the metformin. And we were happy to see that in all of the donors that we tested, we see a decrease in HIV activity. Overall, on average, about 37%. Okay? And so this is consistent with the notion that metformin can increase this protein DDIT4, which will decrease HIV activity. Now, this is all, in a petri dish. This is all kind of these kinds of experiments. But what about cells from actual people living with HIV? And so to test this, we worked with our collaborators from UNC, in particular Ashok, who's in Ed Brown's lab, also with my close colleague, Nancy Archer, who is also at UNC. And we took cells from 4 ART-suppressed people with HIV. And when you take cells from ART-suppressed people with HIV and you put them into a petri dish, those cells will reactivate, and you can look at HIV activity. And so what we did was we looked at that activity under conditions where we did not add metformin versus under conditions where we did add metformin. And what we found was in all 4 individuals, when we had metformin, HIV activity was suppressed, you can see, in all 4 of these instances. And when you average it all out, overall, it was suppressed by about 51%. And so this appears to be true both in a completely artificial system where we expose with HIV and add metformin, as well as using cells from actual people with HIV. Okay, and what about in vivo? So we don't really know in vivo, but one thing I want to point out was that there was a study that was completed a few years ago. It's called the LILAC study. And in this study, ART-suppressed people with HIV went on a 12-week course of metformin. And this wasn't because of any postulated role of metformin on the actual reservoir. Metformin, as I mentioned, has many effects. It's thought to potentially reverse exhaustion, decrease inflammation. And so it was being tested in the context of that. And one of the things that was measured in this study is the ratio of cell-associated HIV RNA to cell-associated HIV DNA. And this is a measure, actually, of the activity of HIV. And interestingly, what was found was that in some, but not all of the individuals, there was a decrease after the intervention in terms of the activity of HIV. And so, in extreme cases, participant number 1, as you can see here, the baseline had pretty high levels of HIV activity and it decreased quite a bit, but it wasn't true in all of the individuals. And so right now, what we want to do is we want to get specimens from other clinical trials that have tested metformin to try to understand, do we really see the suppression of HIV activity? But overall, these data suggest that in some people, metformin administration does seem to associate with a decrease in HIV activity. Okay, so essentially, I've shown that different individuals will rebound with different amounts of time, and one of the top predictors of when that individual will rebound is the levels of DDIT4, where the individuals that rebound later, so this better situation here, they tend to have higher levels of DDIT4. The individuals that have faster rebound, they tend to have lower DDIT4. But of course, as a field, we don't want to just delay rebound by a little bit. We want to delay it by quite a bit, right? And so the idea is that we want potentially metformin can shift things over to the right-hand side, but we really want to shift it extremely over to the right-hand side. And as many of you know, there are individuals called post-treatment controllers, which are able to control for really prolonged periods of time upon treatment interruption. And so one question is, post-treatment controllers, do they have different levels of DDIT4? So this is actually not in the paper. This is unpublished data. It's a separate set of studies that we are doing right now to compare post-treatment controllers as compared to non-controllers at the time point preceding antiretroviral treatment interruption. But we were very excited to see that when we did that analysis and looked at CD4 T cells from these individuals, we actually also found that the post-treatment controllers had higher levels of DDIT4 as compared to their non-controller counterparts. So even in these extreme cases, they also tend to have higher levels of DDIT4. Okay, so that's that first gene, DDIT4. That's how we ended up identifying metformin. But as I mentioned, I will also briefly talk about this second gene that we identified. This is a gene called ZNF254. It's actually a crab zinc finger domain protein. Remember that crab thing I showed earlier? It actually belongs to that family. And so we postulated that this gene, just like the DDIT4, could also mediate suppression of HIV. And so Zhishong tested it first using the same kind of system that I showed earlier for the DDIT4. He was able to decrease the levels of this ZNF254, again, by only about 50%. He wasn't able to bring it down to 100%. But under these conditions, again, we also see that the activity of HIV is indeed increased when you partially decrease ZNF254. So consistent with the notion that ZNF254 is also a suppressor. But also really interestingly, what we decided to also do is to mine some public data sets that had previously been generated from elite controllers. So these are individuals that can naturally control HIV in the absence of ART. And we decided to look specifically for ZNF254, this new gene that we identified, in the CD4 T cells of the elite controllers as compared to matched non-controllers, as shown here. And we were very happy to see that in the elite controllers, their CD4 T cells also had higher levels of these ZNF254. So potentially that suggests that one mechanism of elite control may potentially be to have this natural way of silencing HIV in the CD4 T cell, although more work needs to be done to demonstrate that this is actually the case. So in summary, essentially, we were studying what determines when the virus will rebound upon treatment interruption. And we identified these two interesting genes. One of them pointed to metformin as a possible way to potentially delay rebound, but you can also imagine that there are other ways as well. For example, if we can somehow specifically deliver ZNF254 into those active reservoir cells, we could potentially silence them in the long term. And so the idea here is metformin could increase DDIT4, which leads to HIV control. Something here, maybe delivering ZNF254 directly could also, silence HIV, leading to HIV control. But I also want to mention that, beyond kind of this notion of treatment interruption leading to HIV control, even implementation of these factors during ART could have beneficial effects, because the ability of HIV to be very active, even during ART suppression, can contribute to the chronic inflammation and comorbidities. And so we are also interested in just understanding whether overall by silencing HIV, we can also improve the overall health of the immune system. And so I will end there. I'll end with this image just because I like to, think of it as kind of representing what we are doing right now. I want to thank the folks that did the work. I acknowledge them along the way, but special thanks to all the participants without whom these studies would not have been possible. And with that, I'm happy to take any questions.
Yeah, that was great, great, great. Very interesting. It seems like you've got a surrogate, you've got, a drug that's cheap, that's easy to deal with. So before you talk about what your plans are, I'm wondering, there are a lot of people in the country in this country alone that have diabetes that take metformin.
Yeah.
So in other words, we're always talking about people that are started at, drugs early, that are early in the game, that are not, the old timers who need help the most. So, I know it's a really bad time to think about something like this, but and that we'd have no healthcare system to be able to look at this. But maybe in the Veterans Administration or something, we could look at people that have been on antivirals for a really long time that are taking metformin to see what's happening with them. Is a kind of in other words, we're always screaming about what about these people, So it's also a way to look at that. I mean, you have a lot of important things going on. And what are your plans for the future?
Yeah, great point. So I talked about, there are clinical trials in people with HIV, and those are clinical trials, not necessarily in people with diabetes, right? Just to assess the effect of metformin. But there's a lot of people, as you mentioned, Linda, that because of type 2 diabetes had gone on metformin, but there are also those that haven't. And so one thing we're planning on doing is leveraging some of these huge cohorts like the Maximize combined cohort study to look in their database to see if we can find matched individuals, let's say both with type 2 diabetes, because otherwise it'd be hard to, get historic samples of people that were on metformin, but those that were versus were not on metformin. Or maybe individuals that had type 2 diabetes before versus after they initiated metformin. So, we're going to initiate to try to look at those specimens, but we need to interrogate this further. And yeah, we very much want to do that.
Okay, great. Michael, you want to go for the chat? There's a few questions.
Yes. So Rafe was asking a question about he saw that there was an increase in activity in participant 6 in the metformin study. Does this indicate that some other confounding variable was occurring?
Yeah, and that's why we want to look at this further, right? So there was definitely variability in that LILAC trial. This was not our study, but the way that the HIV activity was monitored, this study wasn't focused on HIV activity. I just pulled that out because that's what we're interested in. We have better ways right now to look at HIV activity, and so what we want to do is with these specimens that I mentioned that we want to get, we want to look more precisely at HIV activity using a lot of the newer tools that we have, but. Yeah, I think, that's an important point that some individuals may decrease a lot, but some individuals, may not, or maybe even go up. We need to understand why that is the case.
The other question was answered, but there was a question about what milligrams of metformin was being used
in the clinical trial.
Yeah.
Right? Yeah, I don't know. That wasn't my study. Yeah, unfortunately,
Well, and Richard Jeffries kind of answered it in the chat there.
Okay,
so
great.
Yeah. Anyway, let me see. There was just another question that just came in. Let me see.
Patricia had a question about the metabolism of metformin, and could there be individual variation, and what is the actual metformin level? What is the actual level of metformin in the cells intracellularly?
Yeah, absolutely. I mean, I think, and that's where, we can apply some of these like sophisticated assays to look at the residual level of metformin, that's circulating the blood, but also within actual cells, right? And so I think that hasn't been looked at, to my knowledge, in the context of ART suppressed people with HIV, but I think these are all important questions.
Moses Supercharger had a question about what are the effects of all the cuts on this. So it's essentially, to me, that means
I missed that. Effects on
what might be the effect on your research of all the cuts that we're experiencing?
Yeah.
So the work that you need to do to test all this stuff, is it really high-end stuff that costs a fortune and is very labor intensive, or is it, you know what I mean? I mean, it's just things that are scalable that everybody can do?
Yeah, I mean, there's both levels, right? So I think what would be amazing, but would entail a lot of funds and efforts, is to actually do a clinical trial where individuals go on metformin, and then we first monitor to see that the active reservoir does decrease, and then we take them off of treatment, right? Analytical treatment interruption trials. And as I think everyone knows here, those are really, really involved and they're expensive. So, that would be very involved. But, as we were discussing earlier, there are specimens already, existing from people that had taken metformin because of diabetes or other reasons. So I think in the meantime, we can analyze those specimens, and that's what we are trying to do.
I mean, you could really follow people who are randomize them essentially. I don't know how you'd randomize them, but you could just follow people that are on it and are not on it and see what happens.
Yeah.
I mean, that could take a long time, but you could get, I mean, you wouldn't have to do a treatment interruption. You'd get enough, you'd have enough power to do a lot of things, because a lot of people don't want to do treatment interruptions.
Yeah. And we can, we can monitor HIV activity without undergoing treatment interruption. And actually, one of the drivers of that Biomark study, why we did this study, and this was already pre-existing samples, like the Biomark study did not do any new treatment interruption, but was to, really try to identify biomarkers of when someone would rebound, because it's not easy to take someone off of treatment, right?
Right. I mean, the other thing is, I really love the idea of giving this with antivirals or something else. I mean, the idea that we have all these combinations. If we've learned nothing, then you're gonna give one or two interventions, and that's gonna make a difference. Do you know what I mean? I mean, the immune system might need a little bit more help, especially in people with chronic HIV.
Yeah, our hope is that, even if the individuals stay on ART the whole time, just decreasing the active reservoir by silencing it, even
Yeah.
partially, right? Not 100%, even partially, that can boost the immune system. And in theory, a healthier immune system can have many, many beneficial effects. So,
And then if you block it, and then how would you lock it and keep it, inactive? Do you
yeah,
have to?
that's what the HOPE Collaboratory is studying, all the crab stuff. Yeah. So.
Yeah, Patricia has also asked a couple of questions. What will it look like in the future, and do we know if it can lower inflammation?
Yeah, so those are the questions that we're interested in testing. I mean, in general, metformin is associated with decreased inflammation, right? So there could be effects actually completely independent of effects on the actual active reservoir. So it could have, beneficial effects from multiple angles, but
Right.
this needs to be interrogated.
Yeah, I think so, especially in long-term survivors who are, that's the
Uh,
driver of all the bad stuff happening, right? It's not their
yeah.
HIV, it's everything
I
else.
mean,
So.
my sister calls metformin a fountain of youth drug because it does so many good things.
Is that your secret, Linda?
No, I don't have diabetes.
I don't know anything about metformin. Is it a totally chemical? What's the source? Is like was it?
Yeah, I have that slide that I borrowed it from Melanie. It was identical. Let me see. I think it's on that slide how it was. It's from this plant called Gaulega offensiva.
Okay, so it's a plant extract. Okay.
But it's probably just a chemical
Yeah,
derivative,
it's a
right?
compound. Yeah, there's a certain
Yeah.
structure. Yeah, yeah.
So there's
Interesting
a
question.
question. So speaking of the last gift, would you envision testing an intervention like metformin at the end of life with all the ethics, of course?
Absolutely. I know like Davy Smith is talking a lot about like interventions at the end of life. And metformin is a very safe drug, right? There's so much info on it. And so I think, it would be a great one to test, actually. That's a great idea. Yeah.
Mm. And we just have another comment from someone who's on metformin here on the call, and they said it's the most helpful drug they've ever taken. And maybe not ever, they say they have taken.
Hmm.
Yeah, I'll just add, I mean, I have ME/CFS, fibromyalgia, and then I experienced long COVID, and they
Sorry.
all conditions improved with metformin.
Huh.
Wow.
That's interesting.
We should put it in the water.
Any other questions?
Jeff, Michael, Richard. So you're going to do other stuff. Well, what's the actual plan that you're going to do? What first and what next, or a couple at the same time? Are you going to work with groups like the HIV, the ACTG, or other groups? Because, we're supposed to be basic science and somebody else is supposed to do clinical. Do you have any industry partners or?
Yeah, so I would say, two main things with regards to metformin. One is, well, we already have some specimens from individuals before versus after they went on metformin, people with HIV. We're trying to get more of it, including leveraging Max White's combined cohort study. And so they're to really try to understand the mechanism and is it silencing it in all kinds of reservoir cells, just some of them? Why would it only be some of them? Really just to have a better mechanistic understanding. At the same time, with others in the HOPE Collaboratory, we are doing preclinical models in metformin using, animal models to see what is the effect of metformin, not only on the reservoir, but also just on, chronic inflammation, immune responses, et cetera. So I think, we're definitely interested in moving forward with that. And also, there are metformin-like compounds that aren't exactly like metformin, right? Like, would some of them be even better? We're also kind of exploring what other kinds of related compounds one might test that relates to this whole pathway.
Yeah, because metformin can be pretty bad with diarrhea, although it's very safe and does so
Yeah.
many good things. It does have its issues.
Nadia, this was fabulous. I wanna echo in the chat. There's people thanking you for this exciting research, and I wanted to, if people are interested about the Last Gift cohort, the Last Gift movie, want to do a plug because we will have a live panel next week on the 14th at 1 p.m. Pacific time with members of the Last Gift cohort team. Jeff will be on there, Sarah will be on there, but Susan, Jim's wife, will be on there, and we'll have some discussions about the Last Gift study as a whole, but also mostly the human connection. And yeah, it's really great, Nadia. Thanks for sharing all that and all the incredible work that you're doing with the samples and with all the collaboratories. I want to also do a plug for Raif, who's here on the call, because Raif will be doing an interview with Nadia, and that interview will be also visible on the channel. And he has actually an interview with Suzanne Valente, who's another HOPE investigator, on some work that she has done recently. So if folks are interested, this is also something you can have access to, and you can go onto Raif's YouTube channel for all the folks here. Raif, if you want to put that in the chat, people can also learn. Because
maybe once they're all ready, Rafe, you can send them to Michael, and we could send all that to everybody that's been on the call today. That would be a great way to disseminate all your good work. Same with the link for the Last Gift thing.
Yeah, Raif put in the chat that the interview with Suzanne is on spironolactone.
That's great. Okay. Also, if we're gonna do one last thing, besides thanking Nadia for that, was just really fabulous. I mean, we were really fascinated by this and all the possibilities of how far this has come so quickly, because it's a drug like metformin that we're so familiar with. It's so safe, and people may be, we're always telling investigators, your rival is one pill once a day. You can't expect people to get on these trials, do all this crazy stuff, do ATIs, when you're going to need 200 people for a trial. It's going to be a real hard road to hoe. But anyway, we're also going to have, again, on June the 16th, we're really excited about this. We were going to do this as just for the MDCs, but we decided after we got some news yesterday that we're going to just open it up for everybody, and we're going to show, just a giant overview from Richard Jeffries, who collects all this data, who's our what I always call him, our Encyclopedia Britannica, to let us know what's up with everything in all of the different strategies.
Oh, he's amazing.
Yeah, it's gonna be really fabulous. And look, very satisfied, 100%. That's the first time that's ever happened, Nadia. What did we have? 80 last time? It's the highest, 100%. So you get a gold star. I never thought
nice.
you could finish it all. You got 10 minutes to spare to get to your next call. So thank you for this wonderful presentation. Thank you for everybody from DARE, who DARE, from HOPE, who really helped make this possible. And hopefully we'll see you on June 16th. Michael will send out a flyer with info, and Rafe will get all his info out for all the good work he's doing.
Yeah, and join us next week for the Last Gift panel live. If you're around, we'll send a reminder again. I think Sydney fixed the registration issues that Jeff and Michael might have experienced. And Andy is on. I see Andy there also. Was with us at the showing of the movie at the film festival.
Yeah. Michael and Richard can send it to their lists too. So, we're about collaboration and making sure
Absolutely.
Everything we can give and share with other people. So
yeah, now I see Korean is on here as well. Yeah, it's wonderful.
Okay, well, why don't we, since Nadia needs to go, maybe we'll give her time to get a glass of water.
Yes, thanks everybody. Thanks.
All right, thanks so much.
Thanks, terrific.
Bye-bye.
Thank you all.
Thank you. That was
Michael.
awesome.
Bye-bye.